Hello, good morning everyone. My name is Mukundan Kumar and my matrix number is 188644. Today I would like to talk about my project and my project entitled Microalgae Protein Extraction via Subcritical Water. My supervisor is Dr. Razif bin Harun and my coordinator is Dr. Tinia Izati. Now let us move on to the introduction. There are total of 3 different key points in my title, and one of the key points is microalgae. Microalgae is a type of unicellular photosynthetic microorganism which can be observed under a microscope. They are rich in biochemical products such as carbohydrates, proteins, and lipids which are produced intracellularly within the cell. The advantage of using microalgae is that it has high growth rate, it is easy to grow, and it has high production capacity of protein. Microalgae that is being used in my project is Nanochloropsis species. Now let us move on to the second key point, which is the protein extraction. We can extract out protein from various types of protein sources such as fish meat, de-oiled rice bran, hog hair, eggs, and also soya bean. Microalgae is considered as one of the viable source of protein. This is because microalgae has higher protein yield per unit area compared to the terrestrial crops. Next key point will be the subcritical water extraction. Subcritical water extraction is one of the emerging technology. It is also known as green processing technology because the solvent that is being used is water. It is performed within a temperature range of 100 degrees Celsius to 374 degrees Celsius and kept under an adequate pressure in order to maintain it in a liquid state. The advantage of using subcritical water extraction is that it is cleaner, it takes shorter production duration time, and it is cheaper compared to the other conventional method. Now let us move on to the problem statement. Chemical-based solvent has been used during the conventional methods. Using chemical-based solvent towards food-based product is very harmful because those chemical-based solvent consist of toxic materials which can harm the human health. It has to undergo several ultrafiltration and purification in order to remove all the chemicals substances from the food-based product. It also requires high amount of money and sophisticated technology to remove all the toxic substances from the food-based product. Next, we'll move on to the objective where I have 2 different objectives in my project. One of the objectives is to investigate factors such as the temperature, time, and microalgae loading that affects the composition of amino acid production from microalgae Nanochloropsis species via subcritical water. My second objective is to understand the kinetics and the thermodynamics of amino acid production from microalgae via subcritical water technology. In my project, I have given 2 hypotheses, and one of the hypotheses is that the temperature plays a vital factor that affects the extraction of amino acid from microalgae. The second hypothesis is that the subcritical water extraction is one of the ideal methods to extract protein from microalgae. Now I will brief about the methodology of my project. There are a total of 3 sets of conventional experiments that will be carried out, which is based on the 3 different factors, which are the temperature, time, and biomass loading. At the end of the 3 sets of conventional experiments, we will obtain the optimum temperature, time, and biomass loading for the highest protein yield. In the first run, temperature will be varied. As you can see here, there are 5 sets of temperature that will be tested during this conventional method. And the biomass and also the time is kept constant, where the time is kept at 5 minutes, whereas for the biomass loading it is kept at 5%. At the end of this conventional method, the conventional experiment, the temperature that gives the highest yield protein will be used for the testing of the second parameter, and the second parameter will be the reaction time. In this reaction time, we can see that there are 5 different sets of time that will be tested during this conventional experiment, whereas the biomass loading will be kept constant, which is 5%, and the temperature also will be kept constant for the 5 months, and the temperature will be obtained from the previous test. From this conventional experiment, we can obtain the optimum temperature, optimum time it requires to produce the high yield of amino acid. Next, we'll move on to the third set of conventional experiment, which is related to the biomass loading. In this experiment, temperature and time are kept constant, where the temperature is obtained from the first conventional set, and the time will be obtained for the second conventional experiment. But the thing that is gonna be varied will be the biomass loading, where there will be 5 sets of runs will be tested in this parameter. For each set, one of the set is starting from the 5% biomass loading, 8% biomass loading, 10% biomass loading, 15% biomass loading, and finally 20% of biomass loading. From this set of conventional experiment, we can obtain the optimum biomass loading that required to produce the highest yield of amino acid from our microalgae. Next, I will talk about some steps how to carry out this project. The first step will be the microalgae is weighed based on the biomass loading, and a known volume of molecule water is added till 5 ml. The second step is that the microalgae and water are mixed together and transferred into the reactor and being purged with the argon gas. The reason why they are being purged, the reason why the reactor are being purged with argon gas is that we have to remove the excess air inside the reactor. The reactor is then immersed inside a salt bath that has been heated to the desired temperature. The reaction time is then recorded, and once it is completed, the reactor is then quenched inside a cooling water. The reason why it is quenched in the cooling water is that we want to stop the reaction. The next step is that the extract is then poured in the centrifuge tube and being centrifuged. And finally, the supernatant is then decanted and stored at 4 degrees Celsius for further analysis. The product analysis will be done by using UPLC. UPLC stands for Ultra Performance Liquid Chromatography. Only the extracted liquid will be tested in UPLC. The result that we have obtained from the UPLC will be then compared with the acid hydrolysis analysis. The next thing that I would like to emphasize is about the kinetic study. By using the Arrhenius equation, the thermodynamic properties such as the activation energy will be obtained for different set of temperature. And finally, I would like to talk about my expected outcome of this project. There are 2 expected outcomes that I have written down, and one of the expected outcomes is about the temperature will be the most important factor in obtaining the highest amino acid yield of Nanochloropsis species microalgae. The second expected outcome that I'm expecting is that subcritical water extraction is one of the emerging technologies, and therefore the possibility of obtaining high yield of amino acid from the microalgae will be massive. I think that's all from me. Thank you.